Hello everyone, my name is Eric Torres and I'm a marketing manager at Biotium. And today I would like to talk to you about our line of mix and stain antibody labeling kits. So these kits are useful for anyone who's using direct detection of antibodies in the lab. So for those wondering if these kits can be applied to your experiment, let's briefly review the differences between indirect and direct detection. We're most commonly familiar with indirect antibody detection, which employs a primary and a secondary antibody for amplification. The advantage of this technique is generally that secondary antibodies are less expensive. They offer an amplified signal because multiple secondary antibodies will combine to the primary antibody. They offer a bit more flexibility because you can mix and match different secondary antibodies of different labels for different host species. But there are some disadvantages in that the protocols are a little bit longer because you have to add a secondary antibody incubation step, and they tend to have more background than direct detection. This is because uh, there's some cross-reactivity of species when using secondary antibodies, and they may potentially pick up any endogenous immunoglobulins that are present in your sample. For direct detection, the advantages would be, and this involves a primary antibody that's directly labeled. Um, the advantages of this technique would be generally a simple protocol because you don't have a secondary antibody incubation step. You'll have less background because you don't have a cross-reactivity from the secondary antibody. It offers a different kind of flexibility, but not the same kind as indirect detection, in that you're actually able to use multiple antibodies that come from the same host. There are some disadvantages with direct detection in that generally these directly conjugated primary antibodies are more expensive and can be a little bit more difficult to find, um, depending on what target you're looking for. There isn't any amplification from the secondary antibody, and so this direct detection should be used on targets that are generally well expressed, and therefore they're less sensitive. So for those who are looking for the advantages of direct detection, antibody labeling kits can be quite useful because you can label your own primary antibody in-house with any label that you need for your experiment. So for all the antibody labeling kits you can choose from, why choose mix and stain antibody labeling kits? Well, our kits have been engineered and optimized in-house by our research team to really provide superior conjugation efficiency and an optimal workflow for convenience, leading to simple, rapid, and efficient labeling. Not only that, our antibody labeling kits have a patented buffer system that really tolerates a wide range of buffer additives, including BSA, tris, glycerol, trehalose, and other additives. There's 100% recovery of your labeled antibody. There's no purification required, and you don't have to worry about any active dye. And our mix and stain kits are available in a wide range of labels, both fluorescent or enzymatic or fluorescent or chromogenic. This includes 29 of our CF dyes, other commonly used dyes like FITC, Cy3, Cy5, biotin tag or DIG or DNP tag, fluorescent proteins, enzymes, and other labels. And we have kit sizes for labeling any amount of antibody for below 5 micrograms or up to 1 big antibody. And as far as advantages go for our mix and stain kits, I really wanted to focus on the inclusion of our CF dyes for these kits. Now, Biotium is a life science reagent company and we provide a wide range of products, but we take particular pride and focus in developing new fluorescent dyes that offer superior performance. And our line of CF dyes is a strong display of that effort. So these dyes have been validated for numerous applications and have shown strong performance in brightness, photostability, and signal to noise compared to other commercially available fluorescent dyes. And they're available from Blue to AIR. Our mix and stain kits currently offer 29 different CF dyes. Now, I don't have enough time to go into the precise chemical advantages of CF dyes, but we do have a webinar that's dedicated to explaining more on the chemical reasoning about CF dye advantages. That's linked in the description below. It's only 22 minutes, so if anyone is interested in learning more about the chemistry behind our CF dyes, then I encourage you to check out that video. And labeling with our mix and stain kits is really fast and simple, particularly with our CF dye antibody labeling kits. Our protocol is incredibly detailed, but the steps are quite simple. All you have to do is complete pre-labeling checklist of your particular antibody. Then you would add reaction buffer, then transfer it over to the vial with your selected dye, incubate for 30 minutes, and then transfer the entire mixture into another provided vial with our storage buffer. And then you're done. The antibody will be ready. The antibody conjugate will be ready for any downstream staining. So which application you choose for your CF-dye conjugate will be up to you, but our CF-dye antibody conjugates can be used for a wide variety of immunoassays that require fluorescence, such as Western blotting, flow cytometry, immunofluorescence microscopy, but also our CF dyes have been validated for a number of super resolution techniques, including STORM, STED, and other super resolution applications. In addition, because flow cytometry generally prefers direct detection of primary antibodies as opposed to indirect detection, I really wanted to go into the advantages of our CF-dye antibody kits for flow cytometry. Now, as I mentioned previously, our CF dyes are available from blue to near IR in over 30 different colors, but our research team also really focuses on producing spectrally unique dyes that are aimed at filling the spectral gap of fluorophores that are typically used for flow cytometry. And this is all toward helping the design of highly complex panels. And this is particularly relevant with spectral flow cytometry, which is now able to accommodate 40 color panels. And so our dyes have actually been published with the help by SciTech in these types of panels, and actually shown to really pave the way for producing highly complex panels because of our spectrally unique dyes. And not only that, our CF dye antibody conjugates have actually shown higher stain index and signal to noise over other alternative fluorophores. And so this similarity index table you see here is all data that was obtained, that was provided by SciTech, and it just shows a number of our CF dyes that are suitable for spectral flow cytometry. And it just shows the wide range of multiplexing options our customers have if they use CF dyes. And we believe that better conjugates come from better dyes, and thus our CF dyes really offer advantages for our mix and stain kits. And you can see that in the data. In this comparison of cells that were labeled with a endosome and plasma membrane marker, the antibody that was conjugated with a CF dye through mix and stain performs much brighter and shows much more specific staining than a spectrally similar dye that was labeled with the competitor lightning link kit. And as I mentioned previously, our mix and stain kits are actually able to tolerate a wide range of buffer additives that are present in your antibody solution. This is just an example of an indirect detection of tubulin in HeLa cells using a goat anti-mouse CF48 antibody. The staining is actually very comparable toward direct detection of the tubulin antibody, which was labeled with mix and stain in the presence of excess PSA or labeled A sites. And so all these advantages can actually lead to really beautiful images like this one. This is actually an example of rat testes that were labeled with both indirect and direct detection. The indirect detection was performed using a anti-tubulin antibody that was detecting microtubules in green using a secondary antibody, and the direct detection was performed using a mix and stain labeled tight junctions marker that is labeled here in red, and an active filaments marker that was labeled using a phalloidin conjugate, which is in cyan. So now I'm going to go into the other types of mix and stain kits we offer. We also offer labeling kits for fluorescent proteins that are typically used for flow cytometry. This includes RPE, APC, and PerCP. And remember that our mix and stain kits also offer really high conjugation efficiency, which leads to lower background and higher signal. And you can see that here in the data, where our mix and stain APC conjugation kit compared to a competitor kit really produces much lower background than the competitor kit. For those of you who are looking to label your antibodies with fluorophores that have longer Stokes shifts, we also offer tandem CF dyes, and these kits come in 3 different flavors. We have one for RPE Texas Red channel, RPE Size 5 channel, and the APC Size 7 channel. In addition, Biotium is actually one of the only providers of labeling kits that are optimized for nanobodies, aka camelid, single variable, or VHH domains. And the labeling process is quite simple. It only requires a 30-minute incubation followed by a 5-minute incubation. And these kits are available with your choice of 7 different CF dyes. We also offer mix and stain kits that have been optimized for labeling small ligands, including SnapTag, ClipTag, and HaloTag, with various CF dyes. And the protocol is actually very simple. It only requires a single 30-minute incubation time, and the fluorescent conjugate will be ready to use. And for those of you who are looking to use your antibody conjugate for in vivo imaging, we also offer a separate type of antibody labeling kit called VivoBright. These kits are optimized for labeling antibodies to 4 of our near IR CF dyes, which offer superior tissue penetration, photostability, and brightness, which is ideal for in vivo imaging. These kits also include everything you need to purify and label your antibody, including buffers, ultrafiltration vials, syringe filters, and is suitable for labeling 3 reactions of up to 1 milligram each. Lastly, here is a picture of our company. So if any of you are interested in learning more about our mix and stain kits, our VivoBright kits, our CF dyes, or any of our other technologies, please learn more at www.biotium.com. And thank you for listening.